Sciences, Kakatiya University, Warangal. And he has also completed as an assistant professor at various colleges like Balaji Institute of Pharmaceutical Sciences, Warangal, Swami Ramananda Tirtha Institute of Pharmaceutical Sciences, Nalgonda, and SRL Institute of Pharmaceutical Sciences, Warangal. And he's having 7 national patents on a pharmaceutical composition for treating cancer through nanoemulsions, improved effectiveness of meloxicam through hydrogel formulations, ciprofloxacin hydrochloride hydrogel formulations, preparation of rosuvastatin formulation for treating human oral squamous cell carcinoma, and thermosensitive or thermoreversible hydrogel formulation for sustained antimicrobial action, and isolation and characterization of fluorescent chemical from banana. And the last one is cloud computing system for healthcare. He's also having one international patent from Australia on artificial intelligence based smart detection of lung disease from chest x-ray. And he's also having 5 grants he has received financial assistance from Entrepreneurship Development Institute of India under DST NIMAT. And he has also received financial assistance of 50,000 from Tuberculosis Association of India. And he also received financial assistance of 1 lakh rupees by Science and Engineering Research Board, DST. And 1 lakh, he has also received 1 lakh by Science and Engineering Research Board, DST. And he received 35,000 grant from Telangana Academy Sciences, Hyderabad. He's also having honors like Associate Fellow from Telangana Academy of Sciences, Associate Fellow from Andhra Pradesh Academy of Sciences. He has also worked as an expert in CPCACE. He also received Best Young Researcher Award 2018 from GRABS Educational Charitable Trust, Chennai, Tamil Nadu. He has published 20 research publications, and he's also published 5 books, and he has contributed and organized various conferences, seminars, workshops, and faculty development programs. He has also given presentations in abroad like Japan, Singapore, and Nepal. And he's also having membership in various associations like Indian Pharmaceutical Association, the Society for Biotechnologists, member of the Association of Biotechnology and Pharmacy, Indian Pharmacy Graduate Association, that is IPGA, and Registered Pharmacist of Andhra Pradesh Pharmacy Council, Association of Pharmacy Teachers of India, that is APTI, and the last one is International Journal of Pharma and Biological Sciences. He also acted as an invited speaker for 9 guest lectures. So I have just briefed the profile of sir. Now I request Manish Kumar sir to start your presentation. Over to you, sir. Now I hand over the program to Manish Kumar sir. Over to you, sir.
Thank you, Arinda madam, for your brief introduction about my profile. Thank you. And at the outset, I'd like to thank management of Nalla Narasivadi group of institutions, Dean Challa Krishnamon sir, Chinnala Krishnamon sir, and convener of this FDP program, which includes Dr. Krishnamon sir and Pavan Kumar sir. And I also thank the management and faculty. I hope today's session will be useful information for you in terms of IPR. Sir, can you share the slides, sir?
I made you host, sir. You can share it, sir.
Okay. Okay, sir. Right.
Yes, sir. It is visible, sir.
So today I'm going to discuss regarding few aspects of IPR and then I'm going to shift towards this dimensions of technology transfer. So is my voice is clear, sir? Is it audible?
Yes.
Sir, little louder, sir.
Okay, sir. So the first slide gives the information regarding a brief glance of various IPR forms like patents, trademark, copyright, geographical indication, industrial design, and trade secret. So for the past 3 days, I think you have been going on to these topics regarding patents, trademark, copyright, geographical indications, industrial design, trade secret. So I want to go a little back regarding history of this patents, so historical background, right from the ancient times. So they used to have signatures and paintings and creations. So substance and evidence, and particularly in architecture, paintings, jewelry, dress, etc. They used to mark their impression. So rewards and monopolies granted by emperors, stamping of jewelers, their initials. So initials on jewelry made by them, which is followed by current jewelers, also the local jewelers. They also having their impression on it. So concept of copyright came only after invention of this printing press. So this is regarding the ancient times. Coming to modern times, so modern patents originated in Europe, where European sovereigns commonly awarded letters patent to favored inventors. The first US patent law were enacted by Congress in 1790 as a part of the Constitution. The first patent was granted by the Massachusetts General Court to Samuel Winslow for a novel method of making salt. And the first patent of United States was signed by George Washington on July 31st, 1790. Coming to India, patent system in India was first introduced by British government in 1856. The Indian Patents and Designs Act 1911 was first enacted under the management and controller of patents with a patent term of 14 years. Initially, it was for 14 years. So the Patent Act came into force on 20th April 1972. So initially, Patent Act 1970 provided only the terms 7 years for pharmaceuticals, agrochemicals and food products, and 16 years for other categories. So we'll see what are the current laws of IPR in India. Copyright, we have Copyright Act 1957. Customs, that is intellectual property rights on imported goods enforcement rules 2007. Designs, the Design Act 2007. Design Amendment Rule, that is 2008. Graphical indications, geographical indication of goods, that is Act 1999. Geographical indication of goods rules 2002. Coming to information technology, the Information Technology Act 2000, Information Technology Rules 2000, Cyber Regulations Appellate Tribunal Rules 2000, and Patent Act 1970, which was amended for 3 times, 2005, 2003, and 2006. And Plant Varieties Protection and Farmers' Rights Act 2001, Semiconductor and Integrated Circuits, which was the Layout Design Act 2000, Rules for Semiconductor Integrated Circuits Layout Design Act 2000. And coming to the last, trademarks, the Trademarks Act was enacted in 1909, and and the rules were enacted in 2002. Coming to Convention and Treaty of India with other nations, that is Berne Convention, which was with effect from April 1st, 1928, which was for literary and artistic works, and Geneva Convention, which was effect from February 12th, 1975, and with WIPO, World Intellectual Property Organization, was from May 1st, 1975. Nairobi Treaty, with effect from October 19th, 1983. Convention of Biological Diversity was effect from February 18th, 1994. and with a WTO member and signatory to TRIPS agreement, which was effect from January 1st, 1995, and Paris Convention Act December 7th, 1998, and PCT, that is Patent Cooperation Treaty, with effect from December 7th, 1998, and Budapest Treaty for deposition of microorganisms was with effect from December 17, 2001. So, what is intellectual property? So, intellectual property is an intangible creation of the mind, usually expressed or translated into tangible form, that is assigned certain rights of property. So we can quote the examples like author's name has copyright on a book, a distinctive logo design representing on a soft drink or any uh, edible products, and unique design elements on website, or a novel pharmaceutical dosage from preparation or process, which may be uh, granted and which may be defined as intellectual property. So what IPR includes? As we have seen in the first slide, it includes patents, copyright, trademark, industrial designs, geographical indications, protection of layout design of integrated circuits, and protection of undisclosed information, that is trade secrets. So it refers to the legal ownership by a person, product, or process, which protects the owner against unauthorized copying or imitation. So for what inventions you are going to get type of IPR? Patents are generally given to isolated polynucleic acids, peptides and polypeptides, enzymes, microorganisms, viruses, vectors, antibodies, probes, vaccines, compositions, expression systems, cell lines, plants, seeds, transgenic organisms, method preparation or use of the above, and medical devices. Trademarks are the words, names, or computer icons, graphical designs, multimedia elements, or use of the above. Sometimes trademarks are also given to medical devices because medical device plays a uh, wide scope in IPR in terms of registered design, biochemical, biophysical, or bioelectrochemical apparatus, trade secrets, laboratory notebooks, design workbooks, customer information, documented internal processes on clinical data generated for therapeutic approval, and plant breeders or plant variety rights for propagating and harvesting material from plant varieties, and domain names especially is given for web addresses. So coming to patents, we are going to deal with the patents today. A patent is an exclusive right granted by law to applicants, assignees to make use of and exploit their inventions for limited period of time, that is 20 years from the date of filing. So the patent holder has a legal right to exclude others from commercially exploiting his invention. So he has a legal right to defend his invention. So in return for exclusive rights, the applicant is obliged to disclose the invention to the public in a manner that enables others skilled in the art to replicate his invention. The patent system is designed to balance the interest of applicants and the interest of society. Now, what are the conditions for patentable? So, any invention should be novel or it should be an improved version of the older one. Suppose you want to uh, convert your dosage form into a novel drug delivery system, so that can be patented. So, how it is going to done, I will explain my own patent in the coming slides. Inventiveness: a patent should be inventive. It should be decided on the material contained in unpublished patents. It may be complex or sometimes simple. The inventive steps should be far different from the existing published patent. So last one is usefulness. The invention should be it should have some utility uh, in public interest. If not, those patents cannot be granted. Now we'll see the flowchart of patent filing of patent to the acceptance of patent. So I think you know that when you have an idea in your mind, when you are about to execute it, you can have two ways. One, we immediately the idea can be patented by a provisional specifications. So if you have a basic idea of any reaction or any method, just write it down on a paper and go for provisional patent. Later you can expand it. We have a 12 months of time where we can file provisionals to complete specifications. So if complete specification is not filed within 12 months, your patent will be abandoned. So once you are done with the complete specifications, then you can go for your publication step, which can be published in IPR journal. For every Friday, a new set of applied patents will be displayed on the website of IPR. So from the date of filing, from the date of filing to the 48 months of time, your patent will be open in the IPR journal. So after reaching 48 months, you need to apply with the form 18 for the request for examination. So once you are applying for the request for examination, so your patent will be examined and you'll get a first examination report. So this examination report raises some objections by opponents. So you may get oppositions. So regarding your work, so if you are uh, done with these objections and oppositions, or defending them very well in the hearing sessions with the controller, so you'll get the grant. If you are failed to clear the objections raised by the examiner, you may apply for re-examination. Again, you need to go for hearing process, and if you fail in the hearing process, your patent gets rejected. If your patent is granted, then we need to go for post grant publication, and again we need to go for secondary opposition uh, flow, where any further proceedings, if any, you have to defend them. So if you are done with the hearing process well, finally your patent is accepted. So this is the flowchart of filing to the patent accepted. So whole step is a expensive process. That's the reason many of the scientists and researchers are not compelled to file their works, because each steps is very expensive. If you start from patent filing to the end of getting grant, you have to lose more than 40 to 50,000 of money. So to solve this problem, because many researchers are moving their research into publications, not they're not making their work into a patent. So the government has launched a program called Kapila Initiative, so which was named after our great scientist and president Kalam, the Kalam program for intellectual property literacy and awareness campaign, Kapila for intellectual property literacy and creating patent awareness on 15th October 2020. So the main objective of this initiative is to encourage the filing of different forms of intellectual property rights by higher education institutions. So, so far on the website of Kapila Initiative, that is Ministry of Education, 46,556 users have been registered. So the advantage of registering under this scheme, so the government is going to bear half of your patent charges. So if you are filing two patents, one patent will be borne by the government. So I hope this you need to avail this opportunity that the government of India in filing the patents, because public funded institutions, they have fund to uh, file the patent, and they are ready to pay the fee of patents. But whereas private institutions, uh, which are facing the problem of paying the fee of filing the patents, so this is a good hope for filing the number of patents. And one more scheme for women. So if you go back to the previous slide, and for examining the report, it takes 48 months. But if any woman entrepreneur is filing a patent, so you may get the grant, you may get the examination report within 1 year. So that is the advantage of filing of women entrepreneurs. So we need not wait for 2 to 3 years for your examination report. So this is regarding Kapila initiative. Now you see the statistical database of Indian patents, which was taken from WIPO website. So there were about 45,317 patent applications were filed in India, out of which 12,387 applications were granted, and 29,718 applications were withdrawn or abandoned by the applicants. Maybe the reasons they have not filed the complete specification, so they have stopped their work there itself at uh, initial stage. So about 3,203 patents were rejected on the ground of non-fulfillment of patentability criteria by the invention. That is, they are not novel, non-obviousness, and they don't have any industrial application. So they have been rejected under Patent Act 3 and Section 3 and 4. So what does this Section 2, Section 3 they say? Inventions means a new product or a process which involves an inventing step and having industrial applicability. If you are not fitting into this section, your patent will not be granted. And Section 2JA initiative step means invention should show technical advancement in comparison with the existing knowledge or having economic significance or both, and the invention must not be obvious. And Section 3D, which defines about mobility and non-obviousness. And the last one, Section 3J, regarding cultivation of plants and growing of plants in new methods. So in growing in new methods of existing plant breed is objectionable. So I'll give some examples of few patents which have been rejected by the Patent Office India. So one of the patentee has filed a complete specification with the title powdered formulation for valganciclovir. So it is antiviral drug. So he has prepared a salt form of this antiviral drug, valganciclovir hydrochloride, which shown good acceptable physical, chemical, and light stability at ambient conditions. So the applicant has prepared a dosage form in which he prepared a liquid dosage form for pediatric preparation. But when he has generated the data in his laboratory, it has failed in showing the stability in terms of short-term stability studies were being failed. So immediately he has shifted his work from liquid dosage form to wet granulation method. So where he has added organic acid, which aids the solubility and stability of the drug. So with the wet granulation method, he has prepared uh, granules of this antiviral drug, and his aim is to prepare in the form of dry powder form. So this wet granulation method makes the liquid gets evaporated due to lyophilization process. So those powder can be used as a uh, as a liquid suspension by reconstitution with water. So he has chosen hygroscopic organic acids initially. So they have found that they are degrading the drug. So he has shifted from hygroscopic to non-hygroscopic organic acids. He has chosen fumaric acid, succinic acid, and adipic acid. So he has found promisable results from this uh, acid constituents along with the drug, and he has reported the drug is stable at 3.8, and he has uh, successfully defended the first examination report. But when the controller has called up for hearing, the organic acid which he has used, that is fumaric acid, succinic acid, and adipic acid, are not showing stability below 3.8 pH. So addition with another patent application which is showing another organic acid with which is citric acid, which has showing more stability than these acid which has quoted in his invention. So due to non-technical advancement in his method, so this patent was got rejected. I think this is the reason for rejection of this powder formulation. I think you have followed. The reason is that there was no technical advancement in this. So there was no definite solvent system to adjust the pH below 3.8. So that's the reason the patent got rejected. If you see the second one, patent application for the beta crystalline form of imatinib mesylate salt, which was filed by Novartis but got rejected by the patent office of India. So as per TRIPS agreement, India has started providing product patent after January 1995. So by taking this advantage, Novartis has filed a patent application of pharmaceutically acceptable salt of drug imatinib, and he got the patent in US. And with this confidence, he has applied a beta form of crystalline powder of imatinib mesylate at Indian Patent Office. So Indian Patent Office has rejected the patent. The reason is it doesn't have promising novelty and non-obviousness. So Novartis have argued that why the beta crystalline form is got rejected because it is a polymer form of imatinib mesylate, and it has better flow property, improvement in thermodynamic stability, and it has very good reduced hygroscopicity and good bioavailability also. And the Novartis has moved this case towards Supreme Court, but the Supreme Court has declared that beta-crystalline form of imatinib mesylate enhanced the availability of the drug, but it has poor efficacy. So, on the basis of safety of the patients, this imatinib mesylate salt was being rejected. Beta form, other forms may be having good efficacy, but this beta form of imatinib mesylate salt was got rejected. So, in case of public safety and public utility, so this patent got rejected. And the last one is patent application entitled a process for the production of recombinant proteins using carnivorous plants. So in this patent, the applicant claimed that carnivorous plant can be used as medium for production of proteins. So he has identified that some ex-carnivorous plant, he has not mentioned the name of the plant, which has a ability production of proteins which can be used in genetically modifying by transformation of proteins in the digestive secretion. So hence he applied the patent, but it got rejected under the section 3J, cultivation of plants, growing of plants, harvesting of fluid from the trap was considered as a method of method which is against the patent law of India. So, but by this reason, he got rejected. So I have given these three examples. What we need to think about and what which compound or which molecule you need to apply for patent. So coming to my work, that is a pharmaceutical composition for treating cancer through nanoemulsions. So this patent I have filed on 22nd 12th 2015, and it got published in IPR journal on 23rd June 2017. So I have waited for 48 months. Recently, I have cleared the first examination report, and this work has uh, resulted a grant for me. So the the compounds, the chemical entities, what I have taken in this formulation are rosuvastatin and ketorolac. So rosuvastatin is a known drug which is generally used for, which is used as an anti-lipidemic agent, and ketorolac is used as analgesic and anti-inflammatory drug. So before starting this work, I used to think that how can we use this chemical entity for cancer because they have already proved activity. So by referring few journals and with the help of my friends who are very much well in chemistry knowledge. So we have compared various chemical structures of cancer drugs with the structure of both ketorolac and rosuvastatin, and we have found some moiety which is very much resembling the cancer drugs. And we have also found from the journal source that somewhere some percentage of these drugs are showing chemotherapeutic activity against in cancer treatment. So this evidence made us to work out on this project of preparing nanoemulsion. So we have already existing dosage form of tablet form of this rosuvastatin and ketorolac in the market. So we want to shift our work to a nano method or new novel method, that is nanoemulsion. So the year of 2011 we have initiated this work, and slowly we have uh, gone through BCS classification, solubility studies, and how it is compatible with the various polymers. We need to make it into a sustained release novel drug delivery. So we have gone through the HLB profile of both the drugs, hydrophile and lipophile profile, and we have concluded that we need to start this work. So this was the uh, enhancement or of this work we have extended by applying bioinformatics. By docking studies we have added the work, we have extended the work uh, by docking studies. So the nano emulsions which we have prepared, first we have tested for its evaluation, then we have gone for cytotoxic studies, then the next level we have been using docking studies for proving the activity more firmly. So this was paper published after we have applied the patent and we got the first examination report. So coming to the explanation of this work, so we have prepared the nano emulsion by using high-speed homogenizer and followed by ultrasonication method. So that is an advanced technique in preparation of nano emulsions. So we have used the probe sonicator for that. And initial studies we have gone through the detection of analytical part, that is measuring the lambda max of rosuvastatin, which was found at 243 nanometers and ketorolac at 320 nanometers, and we have chosen the solvent system for this formulation is 65 and 35, that is 65 ml of methanol and 35 ml of water. So we have developed a method with a standard drug of rosuvastatin and ketorolac separately, and we have got good calibration curve showing good RSD values less than 2%, which is according to ICH Q2 R1 guidelines. So then we have upgraded the method from UV to HPLC. So same pure drug we have established a method. So then we have gone to the second step of our preparation of nanoemulsions. So we have chosen polyroxomer as polymer, a surfactant, and a labrafil as co-surfactant. And oils we are using methane and caproil we have used the oil. So we have prepared the different formulations by factorial design method. So a series of formulations we have prepared by dissolving 20 mg of rosuvastatin and 350 mg of polyroxomer and 350 mg of labrafil as co-surfactant, and we've added water drop wise, and we have undergone the ultrasonication method for the preparation of stable emulsions. So excess drug we have removed by centrifugation process that we have done in cooling centrifuge, and the drug is removed. So after determining the stability of the emulsion, we have gone for concentration of drug, that is amount of drug present in the formulation by HPLC. The solvent system we have used is 65 to 35 methanol and water. So we have found promising results of drug content in the formulation. So then we have moved towards the evaluation of this nano emulsion. So determination of drug content and determination of viscosity we have done through uh, Brookfield viscometer, and followed by solution state stability by using various pH solvents like 1.2, 4, 6.8, 7, and 7.5. So we have tested our emulsion at various pH levels. Then we have moved towards in vitro uh, release kinetics, which we have done through France diffusion cell, where we have used a dialysis bag, and we have taken the two compartments, that is acceptor compartment, it is 6.8 buffer, and we have tested the amount of drug moving from upper compartment to lower compartment, that is acceptor compartment to donor compartment, uh, at time interval from 10, 20, 30, 45, 60, 90, and 120 minutes. So we have estimated the amount of drug moved from the dialysis fluids through HPLC by the same mobile phase composition. So then we have performed buccal membrane permeability studies by Procane membrane, and followed by HLB hydrophil lipophil balance. Also, we have seen that the scale is 0 to 20, and water in oil it is less than 6, and water oil in water is greater than 8. So our values were around 13.6. So uh, the emulsion was oil in water type. So then we have conducted some bioavailability studies on rat models. We have selected 3 groups, and we have named them as control and test, and we have given the drug with a propylene glycol as a solvent, mixed the drug, and given through oral route. So, and we have collected the blood samples at half an hour, 1, 2, 3, 4, 6, 8, 10, 12, 18, 24, 36, 48, and 72 hours. And we have found the Tmax and Cmax. We have determined the Cmax and Tmax of the drug. The next level we have moved towards the in vitro cytotoxicity studies, where we have sent our samples to Attract Mumbai to perform the cytotoxic studies of this rosuvastatin and ketorolac. So they have performed on K562 cell lines by using MTT assay. So the dye method, which they have performed at 490 nanometers using ELISA reader. So this whole, what I am saying right from the beginning of selection of the drug, converting the traditional method into novel method, and conducting various evaluation tests, followed by uh, testing the activity, and all the steps we are strictly follow. We have followed the ICH guidelines of. If you think about validation parameters, we have followed uh, Q2 R1 guideline, that is validation parameters, and coming to the drug stability and quality risk management, we have seen Q8 and Q9. So, so regarding this, you can ask any questions regarding this work if you have anything before moving to the next slide.
Dear participants, if you have any queries or clarifications, please
Because
interact with sir. Go ahead, sir. Maybe they may ask at the end.
Okay, okay. So, sir, if you have any, because we are moving to the actual topic, that is technology transfer. So, I'm going to shift from this topic to the next level. Shall I, sir?
Yeah, okay.
So, the word technology transfer. Suppose you are doing your work in laboratory and you're filing a patent. So, what is the aim of filing a patent? A question arises. So, frankly speaking, the investment what I have kept on the above the process what I have started, it costs around, only for work I have spent around 60,000, and for filing patent again it costs more than 40,000. So this is a costly affair, I said. So there should be somebody to develop in larger scale or scale up it. If not, there is no purpose of the doing the patent. So during the filing of patent, due to the novelty of this work, some of the agencies have applied me through email. So I have responded to them, and they have found some novelty in this work. And these are the agencies which act as a barrier between manufacturing companies, pharmaceutical companies, and the inventor. So they are like bridge between them. So uh, they'll make agreement which is non-exclusive. We have two types of agreements: exclusive and non-exclusive. Exclusive is nothing but we can't give the work to other persons, uh, other pharmaceutical companies. Whereas non-exclusive we can give. So this is a good example of your COVID vaccine, because India is being adopting the technology transfer agreement. So we are getting Sputnik, other different types of vaccines which are ready to manufacture in India. So this, which is a non-exclusive agreement. So I have signed the agreement with this agency. So I have visited the premises of that agency. I'll let you know the name of the agency at the end of the session. So we need to present our work in detail with the statistical details, and we need to defend the questions raised by the bidders, which are no one but the manufacturers. So once they get satisfied, they'll invest on your work, and you need to transfer your whole rights. So that depends upon three phases, that is licensing term or single agreement, or you can go for MOU. So these three so I'm waiting for the result because I have completed the part of our work. So we are awaiting the result. I hope they respond positively. So this is a brief introduction regarding technology. So technology transfer is defined as the process by which a developer technology with the commercial partner that will. Hello. Hello, can you hear me, sir?
Yes, sir, it is audible.
So in between there was a poor thought the slide has been missing. So technology
Okay.
transfer is defined as a process by which developer technology makes this technology available to the commercial partner that will exploit the technology.
Sir, sorry to disturb you.
So
There
research
is
is
no
carried out.
sharing, sir.
So it's
Your
not.
PPT, your PPT is
How,
not
sir?
visible. Not visible, sir. PPT is not
Can
visible.
you?
Sir, shall I play the PPT here? If you have any technical problem, Manish sir. Hello.
Now is it visible?
No, sir. Please share it again, sir.
Okay, sir. Somebody has disabled, sir. You just enable me. I will.
Well,
It's showing host disabled participants
okay,
screen
okay.
sharing.
I'll check it. Co-host, okay. Yes, sir. I made you co-host, sir. Now you can share. Manish, sir. Manish, sir. Manish, sir. You are the co-host now, and you please unmute yourself, and you can share the PPT, sir.
Is visible, sir, now? Slides are visible.
No, sir. Manish has started the screen sharing. That's it. We are unable to see the slides.
Now, sir, I have changed it now. Again, have reshared it.
Or else, shall I play the PPT for you, sir? Yeah, now it is
You
visible,
play,
sir.
sir.
Now
Okay.
it is visible, sir. Perfect.
Okay.
Please go ahead, sir.
Sir. So the term technology transfer is very important in terms of making your work or your experience in the laboratory, your novelty in the laboratory can be shared to the public. So the problem arises is that how far we can afford in a commercial scale. So scaling up your innovation into public domain is very much a costly affair. So to bridge the gap between the inventor and manufacturer, so this plays a major role. This agreement plays a major role, that is technology transfer. So research is carried out in laboratories on experimental scale, that is laboratory scale, before it will be produced for a commercial use. So transfer of technology is important for such research to materialize on a large scale for commercialization. So you can see the different stages involved in technology transfer. So if you see the invention idea that is made into prototype at your laboratory. So here the work of this inventor stops, because moving towards product development in a pilot manufacturing, that is initial pilot scale manufacturing of the prototype, which is as I said, it involves lots of money. So once you get succeeded there, it will move into for commercialization. So that is called as pharmaceutical development. So once you have developed your formulation or your work in laboratory with successful results, when you can go for commercialization by having a technology transfer agreement between you and manufacturer. So they will take over the work and they will make up into a commercial manufacturing. And if the product fails in pilot stage, the product gets discontinued. So if it succeeds in initial manufacturing stage, they'll go for larger scale for more commercialization. So this is the schematic form of the movement of this technology transfer. So now the transfer of technology could happen between government labs to private sector firms, between private sector firms of the same country, between private sector firms of different country, from academia to private sector firms. So coming to government labs. So So the government labs to private sector. So this type of technology transfer is more advantageous as the government labs can invest and give financial support to the private firms. So where they can uninterruptedly do the research work and technology can be developed by the private sector. So government funding will be there. We have BIRAC scheme by DBT where it promotes the academia and industry together by funding uh, both the areas. And second one is between private sector firms of the same country. So this type of technology transfer generally occurs due to lack of appropriate financial resources or inadequate knowledge of regulatory requirements of the small companies. So that so thus the private sector that develops technology will be absorbed by the high-end manufacturing unit. So that is called as between private sector firms of the same country. So between private sector firms of different countries, so underprivileged countries where they have poor regulatory aspects. So if you are acquiring those technology and if you are implementing in the developing countries like India, so that will be a beneficial to the society. So that is between private sector firms of different country. And the last one is from academia to private sector firms. So in this type of technology transfer of the work will be from the academic institutions by collaboration with the private firms. So where the research researchers from the universities or institutions which are unable to afford, so they can be financed by these private sectors and that can be materialized. And the term technology transfer is not only applies the prototype of work from uh, academic to industry. So we have two types of technology transfer, that is vertical type and horizontal type. So vertical horizontal is nothing but right from the basic. That is, suppose you have, I can give my example that I have changed the formulation from anti-lipidemic activity to anti-cancer activity. So right from the basics, we have changed the entire molecule structure, formulation. We have dosage form is being changed. So right from the basics to the formulation aspects to that is called vertical technology transfer. Horizontal is nothing but shifting of your R&D to the next premises because we have a number of units here. For example, Arvindo has number of units. Dr. Reddy's has number of units within Hyderabad. So shifting the technology within the division of that is different premises. So that is also called as technology transfer, where it involves uh, scaling up from small scale to higher scales. That is called as horizontal shift of technology transfer. So what are the goals of this technology transfer? So the, the main goal of this technology transfer is to enhance product and process knowledge between the manufacturing and within or between manufacturing sites. So it may be within the manufacturing sites or from the prototype developing institutions, whether it is government funded or private funded institutions. So this is the goal of technology transfer. Now it's the objective of this technology transfer to explain the processing information to transfer technology from R&D to production site by listing out information gathered during R&D. So whatever the various information documented during development of existing